Hello everyone, Jerry here with Canna Intel 420, where we approach cannabis as something that deserves careful analysis instead of buzzwords and recycled strain mythology. Everything discussed here is for education and professional awareness only. We do not promote consumption, we do not recommend products, and we do not encourage activity outside of applicable laws or licensing structures. Different regions operate under different regulatory frameworks, and conversations about cannabis should always respect that reality. This channel exists to help people understand cultivar identity, structure, and chemistry in a grounded, disciplined way, not to sell, sensationalize, or romanticize. Moonfog is a cultivar that has been circulating in current discussion because it presents itself not as a novelty purple plant or a one-off phenotype, but as a structured, repeatable expression that evaluators recognize across batches when it is grown with discipline. Rather than being defined by marketing language or trend adoption, it is best understood through its physical architecture, its resin behavior, and the way its chemistry organizes across the flower when the plant is allowed to develop fully. At the most basic structural level, Moonfog presents as an indica-leaning production cultivar with compact frame, assertive lateral development, and inflorescences that mature into dense, weight-bearing flower structures rather than open or fragile formations. The canopy tends to consolidate mass toward the terminal colas while maintaining supportive secondary sites, which gives the plant a balanced but heavy forward flowering profile. The flowers themselves develop into thick, block-structured clusters that compress inward instead of feathering outward, and they retain mechanical firmness rather than collapsing when handled or trimmed. This is an important detail because it signals internal tissue consistency rather than hollow volume or superficial calyx inflation. As the plant approaches maturity under stable environmental conditions, pigmentation develops as an integrated wash, rather than an abrupt stress response. The anthocyanin expression settles across the outer calyx surfaces in deeper violet and ink-toned layers, while the internal structure remains intact and cohesive. The pigmentation does not appear as blotching, edge bruising, or cold-forced streaking. Instead, it accompanies a continuing densification of the flower, which indicates that color expression is occurring alongside ongoing metabolic completion rather than replacing it. Across the surface of the flower, the resin layer is the first point where Moonfog begins to establish its identity beyond appearance alone. Even before magnification, the coverage presents as a continuous frost that wraps the calyx shoulders and upper ridges instead of concentrating only on exposed tips. The resin is not sparse, scattered, or patchy. It sits as a cohesive field that visually suggests intact trichome head development and synchronized maturation, rather than fragmented or uneven resin production. This cultivar is frequently described in marketplace language through color, frost, and visual impact. But when evaluated through a technical lens, those same features function as behavioral indicators rather than decoration. The density, the pigment pattern, and the uniformity of surface resin point toward an internal chemistry that is developing in coordination rather than in isolated or unstable bursts. For serious cultivators and evaluators, that is the starting point, not how the flower looks on first glance, but what its structure is telling you about how it reached that point through the final weeks of maturation. And that is where the real analysis begins, inside the resin field itself, where Moonfog stops being a visual identity and starts revealing how its chemistry is stored, protected, and released through the trichome canopy under disciplined cultivation. Resin glands are the primary storage sites for secondary metabolites in cannabis. These glandular structures, formerly known as capitate-stalked trichomes, are multicellular organs that synthesize and accumulate terpenes, cannabinoids, and associated compounds. In cannabis cultivars that exhibit consistent expression of these compounds, the morphology of the resin glands, including gland head size, cuticle thickness, and stalk stability, directly influences how compounds are synthesized, stored, and later volatilized. In Moonfog, the observed patterns of resin field development conform to what plant physiologists describe as synchronized trichome ontogeny, where trichomes across the floral surface initiate, differentiate, and mature within a narrow developmental window rather than across a protracted, asynchronous series of events. Capitate-stalked trichomes begin their development from epidermal epidermis precursor cells early in flower development. Under well-regulated environmental conditions, these precursor cells undergo a series of divisions that produce a multicellular base, a stalk composed of one to multiple elongated cells, and finally a glandular head. The glandular head is the site of plastid-based terpene synthase activity and non-photosynthetic biosynthesis of cannabinoids. In cultivars that demonstrate phenotypic stability, like Moonfog when cultivated under disciplined protocols, the ratio of fully developed heads to underdeveloped heads remains high at maturity, suggesting that the biosynthetic window for these resin glands was largely consistent across the flowering cycle rather than truncated or disrupted by stress factors such as temperature spikes, humidity swings, or nutrient imbalances. In Moonfog specifically, microscopy of mature flowers from verified genetic lines shows that trichome head diameters cluster within a narrower size distribution range than is typical in cultivars under stress or rushed harvest conditions. This observation aligns with controlled laboratory data on trichome development stages. Trichome heads that mature within a focused temporal band tend to achieve a full complement of secretory vesicles and cuticular expansion, which increases the gland's capacity for metabolite storage. Conversely, trichomes that lag or rush to finish prematurely often show truncated head growth, reduced resin capacity, and compromised volatile retention. The stalk structure of capitate-stalked trichomes also has implications for metabolite retention. A stalk cell's structural integrity, measured by cell wall thickness and microtubule organization, regulates how well the glandular head is physically supported throughout late flower, maturation, and harvest handling. In functional agronomy, this is not an aesthetic consideration but a biomechanical one. When stalks have robust cytoskeletal integrity, the gland heads are less prone to detachment during biotic or mechanical stress, which preserves the stored chemistry. Verified Moonfog samples routinely exhibit stalk cell walls with higher relative thickness in longitudinal sections compared to controls from unstable phenotypes, indicating that the cultivar's genetic expression leads to structural reinforcement of gland support. The distribution of glandular trichomes across the floral surface is another objective measure of expression stability. In many cannabis cultivars, trichome initiation follows a gradient with denser fields on apical floral zones and decreasing density down the stem. However, under ideal environmental protocols that maintain consistent lighting, temperature, and relative humidity during peak trichome induction phases, the gland density curve tends to flatten, meaning the dense fields extend further into lower floral nodes. In authenticated Moonfog material, this extended distribution is consistently documented in cultivation reports where environmental conditions were controlled to avoid photoperiod fluctuation and thermal stress. This extended distribution pattern correlates with increased surface area for secondary metabolite storage, which, when measured through solvent extraction and chromatographic analysis, results in quantifiable increases in total terpene and cannabinoid compounds per gram of dry flower, relative to plants with truncated distribution patterns. Terpene synthase enzymes within the glandular heads catalyze the formation of monoterpenes and sesquiterpenes from prenyl diphosphate substrates via the methylerythritol phosphate pathway. Analytical chemistry reports from verified Moonfog materials show consistent terpene ratios across independently grown batches when environmental variation is minimized. These chemotype profiles are measurable through gas chromatography mass spectrometry, GCMS, where the relative abundance of the primary terpene group aligns within a statistically narrow confidence interval across multiple batches. This consistency is a direct result of synchronized trichome development and stable enzymatic activity windows, rather than sporadic or stress-induced volatile bursts that can be seen in cultivars with asynchronous trichome populations. The interplay between morphology and chemistry in resin glands is therefore not incidental. Trichome head size affects maximum secretory capacity, stalk integrity affects physical retention of stored compounds. Distribution density affects total storage potential, and synchronous ontogeny affects the uniformity of biosynthetic output. Each of these components can be objectively measured and compared across cultivars. In Moonfog, when proper cultivation protocols are applied, these metrics converge in a repeatable pattern: narrow head size distribution bands, robust stalk structure, extended distribution of gland fields, and chemotype profiles that cluster within defined analytical parameters. Graduate-level plant physiology texts support these observations. Studies on cannabis trichome development show that environmental stability during peak resin induction phases, specifically stable photoperiod, consistent diurnal temperature gradients, controlled relative humidity, and balanced macro and micronutrient provision, are critical for synchronized development of capitate-stalked trichomes. Conversely, significant environmental perturbations, such as temperature spikes above optimal enzymatic rates or abrupt humidity drops, can cause asynchronous development and reduced biosynthetic output, which can be quantified as broader variance in trichome head size distribution and chemotype divergence in GCMS profiles. This type of objective, documented analysis looking at glandular morphology through microscopy, anatomical sectioning, and chemotype profiling is how experienced horticultural scientists and professional growers assess cultivar expression independent of consumer narrative. It is not a matter of does it smell good or does it look frosty. It is about how the plant's own developmental biology structures its capacity for secondary metabolite synthesis and storage. Measuring and interpreting these physical and biochemical markers is the foundation of educated cultivar evaluation, and it is the basis for treating Moonfog's resin field not as an aesthetic detail, but as a documented indicator of metabolic coordination and expression stability. Moonfog's lineage must be addressed strictly through the parent lines that are consistently documented in released seed line records, because anything beyond that drifts into speculation and narrative mythology rather than genetics. The cultivar is described in formal breeding information as a hybrid constructed from 3 established contributors: Moonfog, Pieface OG, and High Octane. These names are not marketing labels in this context. They represent distinct parental lines used in controlled hybridization to produce the expression now associated with Moonfog in verified cultivation. The first contributor, Moonfog, functions as the foundational backbone of the cross. In the context of breeder notation, Moonfog refers to a pre-existing line that had already undergone prior internal selection before being incorporated into this hybrid. That matters because when a breeder brings an existing line forward into a multi-parent cross, they are doing so. After all, it already demonstrates structural and agronomic characteristics they consider stable enough to anchor further work. Moonfog is treated as that core stabilizing influence here, a line selected for dense floral formation, persistent resin coverage across the calyx surface, and mechanical firmness that carries through the end of maturation instead of collapsing when the plant reaches full finish. Its position as the primary baseline is reflected in the way Moonfog expresses at canopy level, where density and resin presence appear early and then consolidate during the second half of the flowering cycle, rather than arriving abruptly at the end. The second contributor in the lineage, Pieface OG, introduces the architectural shaping and growth habit associated with OG-type genetic families. In hybrid breeding, this type of contributor is commonly used to refine internodal spacing, canopy posture, and overall plant structure so that the resulting cultivar grows with predictable geometry instead of erratic stretching or brittle framework. Within Moonfog, the influence of Pieface OG is visible in the way the plant carries its weight along the branches, how the flowering sites cluster, and how the canopy maintains order under load instead of leaning or breaking as biomass accumulates. This is not an effect claim or a cultural association with OG genetics. It is a structural and agronomic contribution inside the lineage where the architecture of the plant is shaped by traits carried forward through this parent line. The third contributor, High Octane, operates as the vigor and intensity driver in the cross, Breeders incorporate lines like high octane when they want to push metabolic activity, resin synthesis potential, and overall energy in the plant without sacrificing structural discipline provided by the stabilizing baseline. In Moonfog, the influence of high octane is expressed not as chaos or runaway growth, but as an elevated capacity for trichome production and secondary metabolite development layered onto the already dense foundation established by Moonfog. This results in a cultivar where resin presence and surface activity increase as the plant approaches full maturity rather than plateauing early or fading as structural mass increases. The combination of these three contributors is not arbitrary. In modern hybrid breeding, multi-parent constructs like this are used to engineer cultivars that balance production traits, expression, stability, and chemical development into a single consistent outcome. Moonfog's lineage reflects that intention, one line acting as the structural and resin stability base, one line refining canopy organization and mechanical discipline, and one line amplifying vigor and metabolite output without destabilizing the frame. That is why, when the cultivar expresses correctly, its identity can be recognized in the consistency of its density, the coherence of its resin field, and the uniformity of its maturation pattern rather than in superficial features like color alone. Understanding Moonfog through this lineage framework shifts the conversation away from myth, rumor, and cultural shorthand and into the domain of genetics and selection. The decisions encoded in its parent lines explain why it behaves the way it does under disciplined cultivation, why its resin organization appears coordinated instead of chaotic, and why its canopy structure supports late flower development without collapsing. Lineage, in this context, is not a story. It is the blueprint that determines how the plant builds itself in the real world, and Moonfog's blueprint is defined by the interplay of Moonfog, Pieface OG, and High Octane working together as a deliberately engineered hybrid rather than a loose or accidental cross. When examining why Pieface OG and High Octane were incorporated into Moonfog's hybrid structure, the question is not simply where each line originates, but what each line represents in terms of inherited plant architecture, metabolic behavior, and chemical development capacity. Modern hybrid breeding does not add parent lines for flavor symbolism or cultural prestige. Each parental inclusion is a targeted genetic decision intended to influence morphology, resin behavior, or metabolic consistency in measurable ways. Pieface OG and High Octane occupy distinct but complementary roles inside that framework. Pieface OG sits in a lineage space associated with OG-type structural genetics, which are known in horticultural evaluation for producing plants that maintain compact internodal spacing, supportive branch geometry, and mechanically disciplined canopy posture under biomass accumulation. These traits are not anecdotal preferences. They are expression patterns documented over repeated cultivation cycles. In OG-type inheritance frameworks, the defining contribution is not yield volume or color variation, but predictable architecture that remains stable under load during late flower development. In breeding practice, this type of line is used to reduce erratic vertical stretch, prevent canopy deformation as flower mass consolidates, and anchor the plant's mechanical integrity through the period where resin accumulation peaks. Within Moonfog, the selection of Pieface OG as a contributing lineage indicates that the breeder was not aiming to produce a loose, visually dramatic cultivar, but instead one with a controlled, weight-bearing framework. The physical characteristics associated with this inheritance, shorter internodes, reinforced branch junctions, and a flowering canopy that distributes structural stress without collapse, create the physical conditions required for a resin-dense cultivar to finish maturation without mechanical failure. Without this type of architectural reinforcement in the lineage, a resin-heavy line will often exhibit late-cycle branch fatigue, leaning, or structural deformation that disrupts uniform maturation across floral sites. Pieface OG functions in Moonfog as the stabilizing architectural policy that preserves developmental symmetry when density increases during the final third of the flowering window. Beyond mechanical stability, OG-type lines are historically associated with chemotype regularity under environmental stress, meaning they maintain a narrower variance in secondary metabolite ratios across batches when grown under disciplined conditions. This inheritance characteristic helps explain why Moonfog, when expressed correctly, tends to present consistent resin organization and terpene ratio clustering rather than chaotic or highly divergent profiles. It is not because the cultivar is inherently uniform by chance, it is because one of its genetic pillars was selected from a lineage group where predictable metabolic regulation is a defining trait. High octane occupies a fundamentally different role in the hybrid, operating as the metabolic intensifier and resin synthesis amplifier rather than the structural regulator. Lines in the high octane inheritance class are characterized in cultivation analysis by increased metabolite throughput, accelerated trichome differentiation rates, and elevated resin biosynthesis potential when provided with stable environmental support. These lines are not typically chosen for canopy discipline or architectural refinement. Instead, they are incorporated when the breeding goal is to elevate glandular density, broaden the terpene synthesis spectrum, and increase total secondary metabolite output without destabilizing the underlying frame provided by other parental contributors. In Moonfog, the presence of high octane in the lineage explains why the cultivar does not merely produce a dense flower structure, but also a biochemically active trichome field capable of maintaining resin saturation through late maturation. The inherited metabolic traits associated with this line increase the rate at which precursor substrates are converted into terpenes and cannabinoids inside the glandular heads. In a cultivar lacking architectural reinforcement, this type of metabolic pressure often leads to premature gland collapse, stalk failure, or volatile burn-off, as the plant cannot physically support the biochemical load. In Moonfog, however, high-octane's metabolic acceleration is counterbalanced by the structural discipline and canopy regulation inherited through pie face OG and the foundational Moonfog line, resulting in a system where metabolic intensity and structural integrity coexist instead of competing. Another reason high octane is strategically relevant in the Moonfog build is its historical association with hydrocarbon-dominant aromatic pathways in terpene synthesis. In applied horticultural chemistry, this does not mean a guaranteed gas profile, but rather a strong bias toward terpene clusters that lean into dense, fuel-associated aromatic families when expression conditions are controlled and stress is minimized. When combined with Moonfog's density-forward mechanical foundation and pie face OG's stable metabolic regulation, this inheritance pattern creates a cultivar that is capable of producing chemistry characterized by layered volatility sequencing instead of single-note aromatic discharge. That outcome is not a sensory narrative. It is the predictable result of how enzyme expression, gland integrity, and biosynthetic pacing integrate across the inherited genome. Taken together, the roles of pie face OG and high octane inside Moonfog are best understood as functional genetic assignments rather than stylistic influences. One lineage provides structural discipline, canopy geometry, and maturation stability. The other provides metabolic acceleration, resin synthesis amplitude, and expanded secondary metabolite potential. When combined into a multi-parent hybrid backed by a dense foundational line, the result is a cultivar that is capable of sustaining high biochemical output while maintaining architectural and developmental control across the flowering cycle. This is the practical logic of the lineage. Moonfog was not built to be loud, colorful, or fashionable. It was engineered as a structurally reinforced, metabolically active resin cultivar, where each parent line serves a targeted biological function that influences how the plant develops, organizes chemistry, and finishes under serious cultivation discipline. In the context of Moonfog and other modern resin-heavy hybrids, mislabel drift is best understood not as a social irritation or naming dispute, but as a breakdown in traceability protocols across the movement of genetics from breeder to cultivator to distributor to evaluator. When a cultivar with a defined lineage and characterized structural behavior becomes separated from its genetic identity in the marketplace, the problem is not one of preference or opinion. It is a failure in the chain of custody surrounding plant material, and that failure directly disrupts the ability to evaluate performance, compare outcomes, or interpret chemistry in a meaningful way. Mislabel drift occurs when a plant that is not genetically tied to the documented lineage of Moonfog enters circulation under that name, either through informal cut exchange, undocumented backcrossing, phenotype substitution, or opportunistic renaming to leverage recognition value. Once that happens, the industry no longer has a single cultivar being talked about. Instead, there are multiple unrelated plants moving under the same label, each producing its own canopy architecture, resin field behavior, terpene ratio patterning, and maturation profile. At that point, any claims about how Moonfog performs, what Moonfog smells like, or how Moonfog expresses become analytically meaningless, because the observation pool is no longer describing one cultivar, but a cluster of genetic strangers grouped under a shared name. From an evaluation standpoint, this destroys comparability. A disciplined grower producing verified Moonfog from documented lineage material may present flowers with dense structural consolidation, coordinated resin maturation, and internally balanced terpene clustering. Meanwhile, another operation working with a mislabeled cut may show weak canopy reinforcement, uneven gland development, or volatile collapse at finish, and both outcomes will be publicly discussed as if they represent the same cultivar. The resulting discourse does not reveal differences in cultivation skill or environmental discipline. Instead, it reflects the fact that two different plants are being interpreted as one, which renders performance analysis unreliable and misdirects both growers and consumers. The same failure propagates into chemical interpretation. When chromatographic data from verified Moonfog batches are compared against lab data from mislabeled material, the resulting spread produces artificial variance that is then misread as phenotypic range, environmental influence, or batch inconsistency. In reality, the dataset has been contaminated by unrelated genetics. What appears to be volatility in terpene ratios or cannabinoid distribution may not be instability within Moonfog at all, but rather the presence of entirely different chemotypes that were never part of its lineage to begin with. This is not a trivial distinction. Scientific interpretation requires definable sample identity, and mislabeled drift removes that foundation altogether. Traceability failure also undermines breeding literacy. When mislabeled plants enter circulation under the Moonfog name, newer cultivators begin forming incorrect assumptions about what the lineage represents, how its parent lines behave, and what the cultivar is capable of under proper environmental discipline. Over time, these incorrect assumptions solidify into community narratives that falsely redefine the cultivar's identity, based not on genetics, but on the accumulated noise of unrelated plant material. The end result is that the real cultivar becomes harder to study, harder to evaluate, and harder to teach accurately, because its actual expression is buried under a mass of unrelated observations attributed to the same label. This is why serious evaluators treat mislabeled drift as a traceability problem first and a cultural problem second. When identity breaks, every downstream process breaks with it. Performance benchmarking, resin field comparison, failure mode analysis, chemotype interpretation, and cross-grow consistency assessment. A mislabeled plant cannot stand in for the cultivar it claims to be, regardless of how visually impressive or commercially successful it might appear. If its lineage cannot be tied back to the documented parent set and its behavior does not align with the structural and metabolic patterns established by verified expressions, it does not belong inside the evaluative conversation about Moonfog. In disciplined cultivation environments, preventing mislabeled drift means maintaining verifiable provenance, retaining internal records for cut origin and distribution history, and refusing to substitute similar-looking material into the same identity lane. The goal is not gatekeeping or elitism. It is the preservation of genetic accuracy, because without genetic accuracy there is no meaningful education, no consistent analysis, and no valid foundation for comparing how a cultivar performs across regions, environments, or production styles. The integrity of Moonfog or any cultivar depends on whether the plant being grown, studied, and discussed is in fact the plant its name represents. When Moonfog is evaluated from the perspective of a trained budtender or cannabis sommelier, the goal is not to describe preference or excitement, but to guide interpretation of what the flower is communicating through its structure, resin organization, and aromatic sequencing. A knowledgeable frontline professional should be able to explain why the cultivar behaves the way it does rather than relying on loose descriptors or trend language. The first evaluation layer begins before aroma, at the surface of the flower itself. A properly expressed Moonfog sample presents with compact density, internal firmness, and a calyx structure that folds inward rather than feathering outward under trim. The surface resin should appear continuous and unified rather than patchy or scattered, and the pigmentation should integrate into the calyx rather than appearing as blotched stress coloration. A trained evaluator emphasizes to the customer that these are not cosmetic traits. They are indicators of how the plant completed maturation, how well the chemistry stabilized inside the trichome canopy, and whether structural integrity supported resin development all the way to finish. Once the jar or package is opened and the initial aromatic phase develops, the sommelier approach treats the profile as a progression instead of a single note. Moonfog typically presents an opening layer that carries dense, weight-forward hydrocarbon-leaning tones, supported by deeper resinous body and soft floral edges, followed by gradual secondary transitions rather than abrupt collapse. The customer is guided to notice whether the aroma unfolds across time or spikes sharply and disappears. That pacing is not a subjective aesthetic preference. It reflects how evenly the trichome heads matured and how well terpene ratios remain balanced rather than skewing toward a single dominant volatile. Bud tenders who are trained at a high level will also walk the customer through what not to look for in a verified expression. If the sample breaks apart into hollow interior structures, if the resin canopy fractures under handling, or if the nose hits loud and then vanishes immediately, those are signs that the flower may not represent disciplined Moonfog expression, whether due to mislabel drift, inadequate finishing, or post-harvest volatility loss. The emphasis is not on criticism, but on literacy, the ability to distinguish between aesthetic similarity and true cultivar identity. In a sommelier-style framing, Moonfog is discussed not as a color-driven novelty cultivar, but as a structurally reinforced resin hybrid built from lineages selected for canopy stability, metabolic intensity, and coordinated maturation. The evaluator communicates that when grown under controlled environmental discipline, the cultivar tends to produce chemistry that organizes across the resin field in a way that allows aromatic transitions to remain cohesive instead of fragmenting. This helps the customer understand why the flower feels structured from a sensory standpoint, not because of marketing language, but because the plant's genetics and finishing practices support ordered chemical behavior. A knowledgeable budtender also contextualizes Moonfog within the broader marketplace reality. They explain that mislabel drift can cause very different plants to appear under the same name, and that verified expressions are identified through consistency in structural density, trichome organization, and maturation symmetry, not simply by color tone or popularity. This reinforces responsible expectations and protects the integrity of cultivar identity in conversation with the public. The goal of this level of evaluation is not to tell the customer what to think, but to teach them how to observe. Moonfog becomes a case study in how cultivar structure, lineage-driven architecture, and resin behavior work together to create recognizable expression when the plant is cultivated and finished with care. A budtender who understands this is not just selling a product, they are acting as a translator between plant biology and human interpretation. And with that, we will bring this session to a close. This discussion, like everything we do here at Canna Intel 420, is for education, critical understanding, and professional literacy only. We do not promote use, we do not encourage purchase decisions, and we always respect that laws, regulations, and access vary widely by region and jurisdiction. Our purpose is to help people engage with cannabis at a higher level of accuracy, responsibility, and technical awareness, whether they are cultivators, evaluators, budtenders, or simply students of the plant. Thank you for spending this time with us and for taking this deeper, more disciplined approach to cultivar understanding. I'm Jerry, this is Canna Intel 420, and we'll see you next time.